So for the last talk of this workshop, which I think all of us really appreciated a lot, so everybody was really, I think, proactive, and I could see a lot of discussions, so I was really happy. So for this last talk, we have Alberto Pérez de Alba Ortíz, who is going to talk to us about harnessing machine learning to sample, analyze, and engineer modes in molecular and colloidal systems.
Thank you very much for the kind introduction, and well, thank you very much to the organizer for these fantastic 3 days. And well, it's really a privilege to give the last talk, mainly because I can take advantage of my position and skip many of the explanations that have already been given by the experts. And therefore, I can focus a little bit more on applications and basically on making connections with previous methods. So the work that I'm going to show to you today has been done across 2 groups. One is a group of Ensing in the University of Amsterdam, and the other one is a group of Marín and Dijkstra, which is my current affiliation in the University of Utrecht. And well, these are the people that are involved in the machine learning protocols, but we also apply these protocols to several systems, right? Which we studied with some other collaborators, some of them in Amsterdam and some of them in the University of Barcelona. So the kind of problem that I'm normally interested in is, has already been introduced, which is basically want to go from one state in one free energy valley to a different state going over a free energy barrier. And one thing that we're typically busy with is finding a proper descriptor, a collective variable to bias the sampling, to accelerate the sampling and overcome this barrier. And then once we have descriptors and once we have a trajectory, then these are typically very high dimensional. So we want to understand them to basically extract insight from these complex trajectories. And once that you have some understanding, then you can start to think about engineering these sort of free energy landscapes, like for example, changing the shape of the barrier or changing the relative stability between the systems. And another thing that we are interested in this workshop clearly is coarse graining, which is basically to remove some degrees of freedom in our system. So I'm going to give you some examples of each of these today. I'm mainly going to focus on the first 3 and then just a little bit on the last one. So let's start with biasing. This was done in collaboration with Hooft and Heesch. And I'm also complying with having a picture of a pet in my presentation. So that's nice. And well, typically when we're interested in any kind of transition, so either chemical conformational or phase transition, these are rare events in our simulations, right? And again, we mentioned already the chicken and egg problem, where we're basically trying to find a descriptor for a transition that hasn't yet happened. Like we haven't yet observed the thing that we want to describe. And what we want is collective variables that are well behaved in the sense that they are continuous and differentiable, that you can use them numerically. And we also want them to be low dimensional in order to basically avoid the curse of dimensionality. And on top of that, we also want them to be sensible to structures, dynamics, and free energy. So basically, what are the atomic conformations? How do you move from one conformation to another one? And what are the respective probabilities of each of these transitions? So one approach that we developed that is, of course, heavily inspired in, of course, work done in autoencoders by Andy Ferguson, people who work on genetic algorithms before, like Ma and Diener, and also the concept of the time lag by the final group, and also with low modes like Trajush and Luigi mentioned, is this that I'm showing you today. So here, basically, what we do is that we have a genetic algorithm that chooses collective variables from a pool. This is human chosen pool. But the genetic algorithm basically takes the best collective variables that you can plug into a neural network to try to predict atomic coordinates. And there are several things that you can do with this framework. So for example, once that it has selected some collective variables, you can do sensitivity analysis to rank them according to their importance to the atomic structure. And since we are already using a genetic algorithm, which is basically taking the place of an encoder in an autoencoder framework, then you can also use a second genetic algorithm to take care of optimizing your architecture in the neural network. And finally, the atomic coordinates that we predict could also be time-like, so we can also predict them in the future. So let's apply this to our favorite example, alanine peptide. And here to make things very, very easy, we start with transition path sampling data, which is Monte Carlo in trajectory space. So this is basically as easy as it can get. And yeah, we apply some invariances to our atomic trajectories, and we have a pool of normalized candidate collective variables. And then we run the genetic algorithm, and after only a few generations, we see that 3 collective variables have dominated the population. And these 3 collective variables are, well, 2 of them are the correct ones, so phi and psi, and there's this extra stowaway that is the distance between the 2 oxygen atoms that makes kind of sense if you think about it. And here you could just introduce a penalty to try to, like just penalize that. But another thing that you can do is just introduce the time lag, and then with the time lag, then it's very clear that the two collective variables that are selected are the ones that we expect. And you can observe how the loss of the population decreases very significantly when you have the two right collective variables, and then at some point you actually try to pick up this OO distance, but it's very quickly rejected. So now let's try to do something a little bit more complex. Together with people in Amsterdam, we were studying what we call Hooft transient base pairing in DNA. So normally DNA is in this conformation, so what we call Watson pre-clamping, the canonical structure of DNA that stores static genetic information. But around 1% of our DNA is constantly visiting this alternative conformation that is known as Hooft, in which basically the adenine, which is this two-ring molecule, rotates 180 degrees. So now it's like basically like this. And it's very important to understand this transition because this Hooft base pairing is involved in several biological processes, including DNA replication. So what we did here is we fed into our genetic algorithm the disconnected stable state trajectory. So here you have no transitions in the data, and we try to predict the positions of the atoms of the base pair that is undergoing the transitions, the two neighbors, and then also the phosphorus atoms in the entire DNA chain to have some long-range structure as well. And our candidate collective variables are many. So we have basically several combinations of angles, torsions, and distances of centers of mass near the region of interest or near the transition base pair. We also have very local descriptors that are basically just hydrogen bonds in the transition base pair. And then we have some long-range movements that basically correspond to tilting and rotation of the entire DNA chain. And there are many collective variables that are selected, but we do the sensitivity analysis. Then the first two are basically what you might expect, and also what we got from literature and surveys of PDB. That is that one of them describes precisely this 180-degree rotation. So this basically the rolling, what that we call of the base. And then another one describes what we call base flipping. That is basically a movement in which the adenine goes outside of the double helix. So basically looks like this. And these snapshots are actually recovered from the decoder part of our framework. These are not MD snapshots. So now that you have these two optimal descriptors, you could actually do umbrella sampling or metadynamics or something like that. But actually, one thing that I developed during my PhD, and actually most of my talks are centered on that and not really on machine learning, is this sort of path-based methods that are very similar to the string method developed by Eric. And here, basically, what the new things that we have is that we have like these cyclic paths that are also adaptive and repulsive, so you can explore multiple pathways at the same time. And in this case, basically, what we have is that one of the paths captures the rotation within the double helix of the DNA, and the other one captures the rotation outside of the DNA. And then what we do is that we apply the discovered collective variables not only to the original sequence, but also to variations of the sequence. And I'm not going to show you all the free energies that we get for that, but just like one representative example. And we learned some very important things from these. First of all, we have some important connections with experiments and previous simulations, so we know that the results are solid. But then also there's the fact that one barrier, which is this barrier of the green path, which is the one with flipping, is particularly lower with respect to all the other ones. And after studying this conformation a little bit, we realized that what happens is that the relative flexibility of the DNA is much greater in that particular direction, which means that it's easier to perform the conformational change. And actually the network had already sort of captured that, because if we notice some of the global collective variables, we actually see that in the decoded structures, we see that there's this bending of the DNA that actually reflects that there's something to do with the flexibility during the transition. And we think that this might be very important, for example, with protein DNA complexes, where a protein comes and of course affects the global structure of the DNA sequence. So overall, we have this framework that can very nicely select collective variables, and we have these path-based free energy methods to study transitions. But of course, there's a lot of human input that's still required, right? So you need to come up with this pool of collective variables. So our next project was about trying to avoid that. So this was done by Alba and Heesch. And here, basically, the idea is similar to what Luigi introduced a couple of days ago. So basically, what we do is that we start with all possible features. So we brute force calculate all distances, all angles, all torsions, etc., between atoms of the system, and we gradually filter them. And here you could choose many filters. We in particular choose separation, non-redundancy, and slowness, which you could measure again in several ways. For separation, we use Paredes harmonic linear discriminant analysis, which basically tells us which are the features that best discriminate between two states. Then for non-redundancy, we use the maximum information coefficient, but you could also use something as basic as correlations or something as more elaborate as, for example, amino by Heesch. And then for the slowness, we use CECAM, but then again, you could use many different things. And the only human input that is required for these filters is that at each point, your features are going to be ranked. And then there are going to be plenty of bad features that are not really useful and a few that you can choose. So basically the threshold is the human input here, which is significantly less than a pool of collective variables. And then once that we have the collective variables, we perform these A to B path calculations. So now first example, again, something that you have seen before is signaling. And we have the folded and unfolded state. And if you choose, you calculate all the distances of the system and remove some that really have low variance, then you get that there's a lot of overlap for the folded and unfolded state. So these are really bad descriptors. But after applying the separation filter, then you get that now you have these two nice, relatively well-separated distributions, but you still have over almost 200 collective variables. So this is not practical yet. Now, if we highlight the atoms that are involved in most distances, then we see that they already point to the two beta sheets and the tyrosine that are important for this process. So there's already some insight there. But then again, these are too many to handle. So then what we do is that we measure the redundancy, and out of, let's say, 2 or 3 collective variables that are redundant, we only keep the slowest one according to Tika, and that leads us to, depending where you put your threshold, somewhere between 7 and 10 collective variables. So this you can practically use in a path calculation. Or alternatively, what you can do is that the redundant features, you group them. So everything that is redundant, you assign them to a distance between centers of mass. And then we can get free energy profiles for both the atomistic based and also the coarse grain based collective variables, which are these two variables that are consistent with each other. They are a little bit too high with respect to literature, but this is okay. This calculation hasn't been refined. So these collective variables were just out of stable states. You could refine it with intermediate state data, as many people have shown. But then I'm going to show you a system that is a little bit more complicated. So together with Carmen Rovira in Barcelona, we were studying blue light photoreceptors that are responsive for this sort of photoresponsive movement that you observe that these microorganisms did. So they're basically running away from blue light. And here, what's very fascinating about these photoreceptors is that they show an activation mechanism that starts only with a local change in hydrogen bonding near the active site. So it's something very, very simple. And these can actually derive in extremely short or extremely long signaling time. So ranging from a few seconds to up to an hour, which is a lot in biological time. And well, using basically multi-scale quantum mechanics, classical mechanics calculations, we were able to observe the first stages of the activation and obtain free energies. But then we were also very curious, okay, what happens afterwards? How is the signal transmitted outside of the protein? And for this, we use the filter, this filtering method. And basically, we start with all the features. And here I'm highlighting in green all the atoms that are involved in several collective variables. And these collective variables came from all the angles, all the torsions, and all the distances between alpha carbons. And you see that there's a lot of green in this area of the protein. That's actually the 5th beta strand. And if you do your path free energy calculation, then you observe that near that area, there's this residue, this tryptophan that is flipping out. And this is actually, according also to Fred experiments, what kicks the signal into the next protein. So this actually makes sense according to the biological system. And then, well, we have this framework that in principle you could put many things into. So as I said, in many of the filters, you could plug in extra things. Like, for example, you could plug in the genetic algorithm, but also many of the things that we have mentioned. And well, maybe later we could discuss, because it is our perception that many CV discovery methods are nowadays ready to do some sort of model consensus. So nowadays you can have the sort of these like meta methods or meta models where you can reach some agreement. But now I'm doing well on time, so I'm gonna just comment that we have also been trying to use normalizing flows in Utrecht to study phase transitions. This is work of Stein by Leeuwen. But well, many experts have already talked about normalizing flows, and they understand them way better than us. So I'm gonna just skip this part and go into how do we analyze transition pathways. And this is work done with Bill and Gispen. And Here the problem is that when you look at phase transitions, these are extremely high dimensional, right? Because you need to worry about the individual environment of each of these particles in order to classify them as Sapna was introducing before. And then here basically what we would like to find are order parameters that are sensitive mostly to structure. We don't really care about the rest, but we do care a lot about the local and the global structure of this nucleation. But now, in order to have these transitions, ideally you would like them to be unbiased, right? So you don't want to study the order parameter of a transition that you bias with metadynamics. And one way in which you can obtain these unbiased transitions is with seeding, as Sapna also mentioned. So in order for your seeding to be efficient, you need to find the critical point where your seed has a 50-50 chance of either growing or melting. And this is done sometimes via grid search, which is sort of expensive, but you can do it with what I call quote-unquote active learning, because this is a sort of poor man's version of active learning, where what we do is that we take two extreme points. We take one, hopefully one melts and the other one grows, and then you fit with logistic regression this curve. Now in formal active learning, you would try to look for the most uncertain point, basically the one that you're most curious about, and you would sample that one next. But in the logistic regression, this comes for free. The most uncertain point is naturally in the middle of your fit. So then you sample that one, and you do this again and again and again. And here I'm showing you basically use the convergence. When you do passive, so basically use grid search, and the other two curves represent at least active learning, and you can observe that it's much better. So you can much quicker get a seed.
The 50% point is a single point, right? It's not some one. Then it also has some kind of minimal free energy, or
it's a single point. Here, not worrying too much about the free energy. This is just more about the probability, let's say, which of course is connected to the free energy, but in this case, we just care about the single point that, according to the first fit of the logistic regression, is at 50-50, which is of course a very naive idea, but once that you iterate, it becomes better. And then from this optimal seed, we can do transition path sampling or transition interface sampling to get unbiased trajectories. And once that we have these trajectories, then we would like to classify the different phases that appear during nucleation. And this you can do, or it's traditionally done by using bond order parameters, right? So this is like a standard bond order parameters. But here what we do instead is that we use a neural network classifier. We train it with the both phases, as you might expect, and the inputs are basically a bunch of different order parameters. Now, normally you would just apply softmax in order to extract the probabilities of being in the different phases. But one additional thing that you can do if you want to also detect novel phases that appear is that you can use a Gaussian mixture model. And here basically what happens is that if after giving the order parameters to a neural network, you have a new phase, this phase will be very far away from the Gaussians, the clusters that already correspond to the trained data, right? So you will have a point that is very far away from everything else. And then you know that, okay, this is different. This is not FCC, this is not fluid, this is something new that wasn't in the trainings. So when we apply this to one of our trajectories from seeding and transition path sampling, the way this basically looks is that you have this growing seed, this growing nucleus, and you see the different phases that we're classifying for each particle. And what's very important here, or at least our interpretation, is that you have BCC that doesn't dominate at any point of the nucleation. So it seems to be always way behind the other phases. And there's also, it's colored in red, and you see that there's not really a BCC coating around the nucleus. So this doesn't seem to appear, which is something that people normally assume that happens. Instead, what we see is that at the beginning, the phase that dominates is something that is novel. So it's something that is different from all the other phases, right? Because this is just forming, so it's not really fluid and it's not really solid. And then eventually, after your nucleus grows, then you see that this new phase starts to get away from the volume growth, and then it starts to correspond to the surface growth, which means that now your novel phase is the interface between the nucleus and the fluid. And now what you see that dominates the bulk is basically FCC and also a little bit of HCD. So basically here we have this framework to optimize our seeds and then do some analysis of the different phases that appear. And well, the other thing that I think is important is also to refine our interpretation and also our amount of data from TPS. So just take these conclusions with a pinch of salt. And now I'm going to go into designing engineering landscapes. So here basically our long-term goal in Utrecht is to be able to do bottom-up design of soft materials. So in colloids and polymers, you have these different kinds of interactions that we're mapping there. And these interactions will self-assemble into particular structures that have particular properties. But it's not really clear if you have rules of what interaction takes you to what property. And this is basically the problem, the inverse problem that we're trying to solve. So basically what we want are cost efficient. We want them to be quick design approaches through which we can control the structures that we arrive to. What are the barriers to arrive to these structures, and also the different dynamics. And the approach that we take has three components. So the first component is a model that is basically just a numerical description, a mathematical description of your system, right? So you can have a potential, you can have a time propagator, and some simulation conditions. So here, for example, is an example of a repulsive potential that we use a lot by Tom Truscott. And then we have an optimizer that is basically a numerical scheme to change model parameters in order to maximize a particular behavior. One thing that we like to do is use evolution strategies. So we use mainly covariance matrix adaptation, which basically draws model parameters or design parameters from a Gaussian population. And then according to the performance of each of these models, then we change the shape of this Gaussian in order to draw different design parameters for the next generation. And then it's very important how do you measure the performance. And we do this with a fitness function with respect to a parameter. And machine learning can really help for this. For example, here what we do is that we have a particular target phase, and then we take the diffraction pattern of that target phase. And we plug it into a convolutional neural network classifier, and then the probability of being assigned to the target phase is your fitness function. This is something that was developed by Gabriele Colli for other purposes, but we are using it now for porous materials like this 2D porous material that you see there. And basically it can take you from something that doesn't assemble at all, so something that basically remains fluid, like you see there in the first generation, to something that gradually assembles into a truncated square phase in this case. And you do this basically by changing the design parameters, which in this case are very simple, just temperature and density of the system. You can also try to use free energy as a fitness. And this free energy could in principle be also calculated with machine learning descriptions. And here, what I'm showing you is a very stable colloidal diamond phase with respect to the fluid phase. And this, again, was also optimized across a few generations of evolution, starting with a diamond phase that was very obscure in the free energy landscape. You can also reverse engineer the shape of pathways. So this is closer to what people know as landscape engineering. So in this case, we have a Lerner-Jones system where the transition between FCC and HCP has an intermediate state that has coexistence. But of course, you could also have a pathway from which you start with that doesn't have coexistence at all, where basically you have to entirely melt one phase before growing the other one. And there are many things that you could do with this inverse design approach. Like I mentioned before, Gabriele Colli, who used to be with us in Utrecht, was using it to design quasicrystals, which is a very interesting topic. And nowadays we're trying to inverse design zeolitic materials like this zeolitic nanotube, for example. And well, if you're interested in inverse design, early in 2023, just pending a few details, we will be organizing a workshop together with Thomas Truscott in the Lawrence Center in the Netherlands. And well, just with the final minutes, I will talk about coarse graining just for a little bit. So this is work done by Campos-Villalobos, Giunta, and Susanna Marín. And I'm just starting to dive on the subject. So this is really work done by them entirely. And here, basically, we're interested in mapping a system, for example, something that has a suspension of colloids in polymers to something more simple. So we're trying to coarse grain this, but of course, we still want to retain fine grain-like accuracy. So basically low computational costs. And we want this to be sensible in the sense of having the right structures and then also having the right free energies, the right thermodynamics. But of course, as it was mentioned before, the dynamics will always be a little more complicated to get. So basically, the way we start this is that we take the original system, and then we first of all map it to a system that is basically a binary mixture of two types of hard spheres. But eventually, what we want to reach is a system with just the colloids that interact with an effective multi-body potential. And here we, of course, care about like these many body effects that Cecilia was mentioning just a few minutes ago. And then we basically care about this many body term here. So this is the thing that we want to fit for the many body correction. And the approach is relatively simple. So basically you have training data where you have from the ground truth, so from the, let's say, like high resolution simulation, you take the positions of the colloids and you take this correction for the many body term that you can determine there just like directly. And then we try to fit this with better parameter symmetry functions, in particular, of course, like the 3 and higher degree. And you choose the symmetry functions that are, first of all, most correlated with the quantity that you want to fit. But then, of course, you also want to choose also symmetry functions that are not correlated with themselves from the pool, right? Because you want diversity. And then finally, you can do a fit. So there you can observe how, after adding a few symmetry functions, so when you have like around 6 or so, then you have very good correlation with the quantity that you want to fit, and your error is also going down. And then just to show you some applications of that, first of all, there's the speed up. So here you can see for different size fractions between the colloids and the polymers, you have always good degrees of the speed up. And then you can also calculate properties of systems that were outside of your training set. Here, for example, you see an interface and you can calculate things like, for example, the interferential tension and relates very well to work done with the fine grain simulations. And in Utrecht, we're working on a lot of different systems. So for example, the rods and functionalized nanoparticles that you can also make coarse grain models of. And well, just to conclude, I would like to basically say that we can do all of these things with machine learning. I think that has been clear during the many talks, the very nice talks that we had here. But of course, there are things that you have to always keep in mind, like what is important to you, what are you willing to sacrifice, etc. And with that, well, I just like to thank you for your attention, and of course, all the collaborators, and I'll be happy to take questions.
Questions for Alberto? So it was in your designing section, you mentioned alpha interactions. So how do you deal with that? Because
I mentioned what, sorry?
alpha interactions in designing. Yes.
I didn't understand.
How do you deal with the alpha interaction in the system? Because it's something I mean.
These are not hard. So these systems that we are designing are not hard.
Okay.
Sorry. The ones in coarse graining are, but these ones are.
So in the first part, you were using the Giunta algorithm to select a candidate of descriptors.
Yeah.
Which kind of criteria were you using there, and what was the purpose? So like, it was more for interpretability.
Yeah. So the reason why we use a genetic algorithm and not an encoder is because we wanted to have interpretable collective variables. Now, of course, you could also use an encoder and then do symbolic regression on that and hope that you can interpret it. And the metric to choose the collective variables is how well they predict. So we use mean absolute error on how well they predict either the current time coordinates or the time lag coordinates or both.
Other questions for Alberto? Yes.
Yeah, really nice talk about it. So the when you're talking about the convolutional neural nets, looking at the diffraction patterns.
Yes.
So do you have a gradient by which you can then update the designs, or is just sort of a screening tool as you're moving, say, temperature, pressure, space? I'm sort of wondering, is there sort of a closed loop you can say, I need to change the force field in this particular way or the conditions in this particular way?
Yeah, the convolutional neural network just outputs the fitness, it's just the number, but we do not use any gradients from the network in order to do the design. The optimization. The optimization is handled by this evolutionary algorithm, by CMA. However, we do notice that, for example, if you use something as structural, so at some point we have used the environment similarity by Piaget on this, and then the trajectory of the optimization is completely different. So we think that the convolutional neural network from the diffraction pattern actually picks up some fluctuations that are relevant for the physics of the system. But this is something that we don't yet completely understand.
Great, thank you.
Okay, so I think we can thank Alberto again.